Hi, Sarah. How are you doing today?
Good. How are you?
Good, good, good. So, for those of us that are unfamiliar with EGH, go ahead and just introduce yourself and tell us where you're streaming from right now.
I am. My name's Sarah Basisi, and I'm Tara Sable Atwood's lab manager.
Okay.
And normally I work in the Center for Environment and Human Toxicology,
Right.
but right now I'm at the Emerging Pathogens Institute. I'm working on the coronavirus testing lab that
Mm-hmm.
we have set up here, and I'm the head analyst on our robot that we have here, which you can see behind me.
Right. Yeah. So, what does that robot behind you do?
So it actually extracts the RNA from our samples. So we get
Okay.
samples, whether they're nasal swabs or um, maybe um, throat swab or anything like that. And first, the samples are lysed so that they're inactivated in
Mm-hmm.
a room where we can do biosafety level 3 plus or 2 plus.
Okay.
And then
Is
once
that the
they're
highest level for the
that
non-EGA
that's
folks?
not the highest level.
Okay.
So there's a 3 and a 4 biosafety levels.
Okay.
Um, so this they wear a bunch of PPE
Mm-hmm.
and they lice the samples. And then once they're lysed, they're inactivated. So I couldn't be infected from them. Um,
Oh, okay.
and so once I get them, I can just handle with gloves and lab coat, and I put it on the robot, and the robot extracts the RNA for us.
Gotcha. And the RNA, that's an essential part of the process for COVID-19 testing. Is
Right.
that right?
So the
Okay.
the testing that we do is uses PCR, which is polymerase chain reaction. So
Okay.
it basically measures. So it actually converts the RNA into a DNA copy,
Mm-hmm.
and then it can measure how much of that DNA copy is in the sample,
Mm-hmm.
or I guess not an actual amount, but it can look at whether it's there or not.
Gotcha. Okay. So it sounds like this is some very sophisticated testing. Is this what, so if we're talking about the United States and having lack of testing in the field for most Americans, is what you're doing, is this what the CDC is doing? Or like, how does this compare
Um,
to
so
the
I think
testing
some of
that
the
all of us
testing
need?
we are so to use the PCR, we
Mm-hmm.
use what are called PCR primers, and basically
Okay.
what those are is just little fragments of the DNA sequence,
Okay.
and so that binds to your DNA to be able to actually measure what's in there. It basically makes copies of it, and then you can measure it.
I see.
so so some of the primers that we are using are the CDC primers. We also have another set of primers that are were created by John Lewnicki.
Gotcha. Okay.
So
So then is this what you're doing? Are other universities, across the nation working on similar testing, or do we have something kind of unique and patent pending here at UF?
um, I am not 100% sure. I know the Lewnicki primer set would be just restricted to us, but the CDC primer set is available through scientific companies where anybody could use those.
Gotcha. Okay. So earlier, before we started recording, you mentioned that you're starting to get a lot of samples, for research purposes from multiple sources. So first responders, from UF Health Shands. So, what step in the process are you in this whole kind of cycle of COVID-19 life now?
Um, you mean what step of the process is the robot, or what?
Yeah, well, so we have folks that are ill in the field, or they might be
Oh,
come in
right.
contact with a sample. So the sample then comes to you, and I know right now we're live waiting for a plate of samples. So you run the testing, and then do you find if something's positive or negative, and then what happens? Just kind of walk me
Um,
through the basic process there.
so there's a reporting system, and I'm not really familiar with the reporting system, how that all works.
Okay.
But we are specifically testing non-symptomatic
Mm-hmm.
individuals in our lab. So we're actually a research project and not a
Mm-hmm.
CLIA certified lab.
Okay.
So we are mainly getting non-symptomatic. So any
Gotcha.
positives that do show up, they get reported, and I'm not really familiar as far
Okay.
as that part
Yeah.
goes. the results come off and the supervisors get it and they do all the reporting. So
Sure. Okay, that's fair. But I think probably like most people, none of us were experts in COVID-19 until about 2 months ago, right?
right.
So I heard that you had to quickly learn how to use that robot behind you in a very short period. So maybe you can just share like how your day-to-day work was kind of impacted and what you needed to accomplish in a short time.
Yeah, so we actually this robot was delivered on a Friday a few weeks ago. I forget exactly what day,
Mm-hmm.
but we had this robot's made by Eppendorf,
Okay.
and we
Oh
had
yeah,
a
I
Eppendorf.
can see it on the back. That can is
Yep,
it
we
EPO Motions 5073. It sounds like a good model.
yep. But yeah, so we had an Eppendorf technician come in on Saturday actually, and
Mm-hmm.
we had to get special permission because the Emerging Pathogen Institute is a secure building with security, and
Right.
normally you can't bring any guests in on the weekend, so we had to get special permission for them to be able to come in on a Saturday,
Yep.
and they did all the programming for us, so they can go in and tell it exactly, we want to add this volume at this time, and then we want to take that volume out or whatever,
Mm-hmm.
and when to change pipette tips and when to use the same pipette tip, and the programming is very complicated for that, so they actually did all that for us, but we were here and they ran us through a test run
Uh-huh.
that day, and then the following day, so on Sunday, we came back and the tech was gone, but we, ran tests and tried to get it working properly.
Right. So I imagine the Eppendorf.
Phone call now to pick up the samples. So.
Oh, all right. Perfect timing. We'll just pause here and
Right.
we'll see the live testing as we
A
go.
minute.
Yep.
Okay.
All right. We are back with
Our
the samples.
They come delivered in a box that's flip closed.
Okay.
And the first thing I do.
Who did these come from just now?
So they came from one of our lysis analysts.
Okay.
Her name is Na. I'm not sure her last name,
Mm-hmm.
and I'm not sure what department or lab she's in. But so the first thing that I do is actually centrifuge this plate to make sure that all the liquid is down at the bottom.
All right. Centrifuging the plate. So the plate comes prepared, it looks like. I don't think Sarah can hear me, but that's okay. Looks like she's spinning. So it looks like a plate, it came prepared, so just ready to be spun?
Yes, the plate, comes with all the samples in a lysis buffer. So it's fully inactivated, so it's not infectious anymore.
Gotcha.
And so it has a seal over the top, and you can get droplets on the top of that seal, which is why we centrifuge it before I pull the seal off.
I see. And so did that come from another floor in EPI, or did that come from another third party?
Just down the hall, on the same floor. Oh,
wow. Okay. So this is live and happening right door to door.
This is live and happening right now.
Wow. Okay. Very cool.
I mean, I could actually, the analyst is still down there. She's aliquoting some serum samples to be stored.
Okay.
And so I could show you her once the robot's running.
Oh, sure. Yeah.
She's got all her PPE on. There's a window.
That would be very neat. Okay. So how long does the centrifuge take? All right. So she's opening the robot. Centrifuge takes about, looks like it took about a minute. Sarah's
pulling the seal off the plate.
Taking the seal off the plate. The robot is ready to receive the plate. This is our live sports play-by-play commentating here. She has the pipette in the right hand. All right, so tell us what's happening here. What are you pipetting?
So right now I'm actually pipetting, so our last column of the plate is just a control column.
Oh, okay.
We just put lysis buffer in that in that column.
Okay.
And so I'm going to do that first.
All right.
And then I'm going to add our positive control.
The positive control.
Which I can explain that again in a minute.
So we're setting this up. We're setting the robot to run. So we're going to get live results, right?
Yeah, we'll see it run. The run does take about an hour and a half.
Oh, okay.
So I don't think we'll see the entire run, but you'll see it in action.
I'll have to do a post-recording commentary at the end and say, "Hey, folks, remember those samples we recorded? They were all positive or all negative."
Okay, so this is actually our positive extraction control. So this is used, so this is a sample that we know is positive, where we've actually grown virus in the lab, and it's already sliced. And so we put this on all of our plates so that we can verify that our extraction is working.
And so that is not, you can't catch COVID-19 from that.
No, it's fully lysed, it's inactivated.
Lysed, okay. Is lysed the technical term for not contagious?
Lysed is basically that it's been broken open and can't infect anybody. And one thing we do every time we touch the positive extraction control, we change out our gloves.
Gotcha.
You don't want to get that positive sample.
Oh, right. You don't want to contaminate other experiments.
You'll get false positives.
Right. So do you have a good supply of PPE there in the lab?
We have a lot of gloves, and we have lab coats. So I'm going to put the plate in the back of the robot here.
All right.
And all of my boxes of pipettes, they'll have lids on them, so I'm going to pull those off.
Okay.
The other thing that I do, the last thing before I start running the robot, is I have to add the magnetic beads that actually are the main part of our extraction. I have to add those into a reservoir.
Okay. So those are the beads?
So these are the beads, yep.
They are going into the reservoir. So now you got one plate delivered to you, is that right, just now?
Yep. Yeah, okay. So I got a plate. And so you can see the setup here. The plate is
okay.
all the way back in the back. You can't really see it.
Yep.
And then this has a bunch of different reagents in it, some wash buffers and different things that are used in the extraction process. And then we have 3 boxes of pipette tips that actually move sample and reagents around.
So then how many samples are in one plate?
So we can run up to 88 samples in a plate because we don't want to have space for our negative and our positive controls.
Okay. And
so this plate actually doesn't have 88, it has 62.
All right.
That just was dependent on how many samples received yesterday evening and this morning.
That makes sense. Gotcha. So now everything is programmed to test.
Programmed. I'm going to double check. We always double check that everything is set up properly. And we hit run. And
there we go. Is the robot going to move?
It will start moving in just a moment.
All right. So you're in the lab there, you're running this machine. Are you the one person that knows how to run it?
No, I'm not. There's currently 2 other people that know how to run it.
Oh, there it goes.
Yep. So the first thing it does is it checks the level of liquid in all of our reservoirs there to make sure that we filled it full enough.
Okay. So I can't really see since I'm far away and in a computer. Is there like a pipette on the arm or what's?
So right now it's using just the camera to see.
Oh, to measure the levels. Okay.
To measure the levels. So there's a little camera on there and it's measuring the levels of all the liquid.
So this robot then takes, not you said 90 minutes to run one plate?
So it depends on how many samples we have. If we have 88 samples, which is a full plate, then it takes about an hour and 45 minutes.
Yeah.
If you have the minimum number of samples, which is just 8 samples, as few as we can run, or 8 or less, then it takes about 30 minutes.
Okay. So and then if you do the math, I guess that's where I had a conversation with Tara, and she mentioned that the max you can run is 350 samples a day, and that's just based on the robot limitations.
Right. That basically we can run 4 full plates on the robot, which 88 times 4 is about
right. 320, 350, yeah.
I don't actually know what just happened because it didn't start.
Uh-oh. The robot is camera shy. Come on, Eppendorf. We believe. So now when we're looking at these, what's on the left there? So we have all the different liquids, and you have a testing plate in the middle. There's some silver objects on the left side. What's on the left side?
Right here in the middle?
No, to the left.
This?
Yes.
This is just it's a rack that holds a waste bag for all of our used tips.
Oh, okay.
So
Gotcha.
It's basically the garbage can.
That's the dumpster for the robot. Cool. Yeah, you don't think about that being a self-contained work environment for the robot, just kind of like a NASA spaceship. You gotta build the trash can too. So right now you're troubleshooting what happened.
I'm trying it again. I don't actually know why it aborted the run.
Okay.
So I'm trying again.
We believe. So now, there's a couple other people. All right, starting back over again. So
Okay.
there's a couple other people, so you can have off days. It sounds like, right? You don't have
to. So right now there's 2 other people, and I'm gonna train 2 more tomorrow.
Mm-hmm.
So we'll have I guess that's 5 people that are trained on the robot.
So is this like 24-hour testing, or is it just from
so
business hours, or how? I mean, I know there's some restrictions, of course, being at EPI, so maybe you can.
It's a little outside of business hours, but we're doing 5 days a week, so Monday through Saturday.
Okay.
And we usually start at about 8.
Okay.
And we have shifts where everybody sort of, if we are running 4 full plates in a day, everyone sort of does 2 plates on whatever their job is, whether it's licensing or running the robot.
Gotcha.
Or doing setting up the qPCR. They do 2 plates. And so the shifts vary.
Hey, there's Dr. Basisi. I see you in the reflection. Special guest. We're having software problems.
Okay, I think it should work this time.
All right, there's times charms. All we would see in the video is just it kind of going up and down and
yeah.
Time, right? Yeah. So, all right. So, how long have we been running this machine now?
I guess it's about 3 weeks.
3 weeks. Okay.
Yeah, I think it's about 3 weeks.
And with testing, there's probably not an end in sight, is there?
I don't think we have an end in sight. We just keep coming up with new places to get samples from.
Oh, from Gainesville and.
Right, like we started getting samples from Jacksonville last week.
Oh wow.
At least I think it was last week we started getting samples from Jacksonville.
Okay.
And then tomorrow we are gonna start getting samples from PK Yonge, I think.
Okay, and PK Yonge is.
One of the elementary schools, I believe.
Oh, okay, gotcha.
So yeah, so there's a study with kids. So now it's actually starting to.
Oh, there we go. I see it.
To run. So it's taking liquid from the first reservoir here, which is just a buffer, and it's putting it in the plate, which is back in the back.
Okay.
So adding it to the sample. So we have so the pipette holds 8 pipette tips, and there are eight samples in a column, or eight
Gotcha.
wells in a column of the
Mhm.
plate. So it does one column at a time
Gotcha.
of the plate. And
So that's where all the time comes with,
right, yeah.
multiple
So there's
columns and doing different
it's multiple columns,
liquids.
and so basically it's how this this procedure works is, so this is a bead-based method, and
Okay.
so the lice sample, it adds a buffer in there, and then it
Okay.
adds the beads in, and it
Okay.
mixes up the beads with the sample, and what happens is that the RNA sticks to the beads.
Oh, okay.
And these are magnetic beads. So later you'll see that the robot will actually pick up the plate from the back and set it in the front here, which is actually a magnetic block, and the beads will stick to the magnet. And then
Uh-huh.
it basically just goes through a bunch of washing steps. So it'll add in a wash buffer or ethanol, and it puts it in there, and then it takes it out. And it
Right.
does that multiple times. And then at the very end, it mixes water to the beads,
Okay.
and the RNA would rather be in the water than on the beads. So you get your RNA sample in the water.
Gotcha. So that was how many separate steps there? Like 4 or 5?
So that was,
Hey, took the trash out.
Yep, seven columns, I think, is
Mhm.
what we have on this plate.
Okay.
And so now it's actually, so the beads, which is what I added at the end, and the
Mhm.
reason why we do that is that they'll settle to the bottom.
Okay.
And so it's actually mixing the beads right now to make sure that they're well mixed and it doesn't just give the sample just liquid.
Is the mix just a drop of the ball, or does it actually
So
shake
it just,
it, or?
no, so it sucks liquid up, and then it lets the liquid back out.
Okay.
So it just sort of, in the science world, we call it pipetting up and down.
Okay.
So it pipettes up and down
Gotcha.
multiple times. So
I just have to ask.
now,
I'm limited in my knowledge of lab testing and everything, coming from
right.
behavior analysis and everything. So what you're describing, is this kind of basic sophomore, junior type knowledge? Is this first year, second year grad student, like what you're describing?
I mean, I feel like people learn PCR at least early in college, like in a basic biology class. I think you would learn PCR. You might at least learn about it in high school, but you might not ever do it in high school.
Sure. Okay.
But I would say in college you would be likely to do some PCR if you early in a biology major or something like that.
I see. Okay. So then the robot
you might not learn much about it if you were not a science major.
Right. Yeah.
But so now what it's doing is it's taking the beads and it's putting it into the sample, and then it mixes the sample up and down to make sure that the beads are well mixed with the sample.
Oh, that's what it's doing right now.
That's what it's doing right now.
That's the mixing. Okay, I see.
Yeah.
So then with this many samples, this will take like an hour to get through all of them. Is that about right?
Yeah, to get through all of the steps, it'll take about an hour to an hour and 20 minutes to go through all of the steps.
Gotcha. Okay. So that's running. So are there any specific like responsibilities related to this COVID-19 testing that you do like while the robot is running, or?
So there's some responsibilities actually with the robot. So
of course, yeah, when the software program doesn't work.
Right, with the software program doesn't work, but as you see, we have 3 boxes of pipette tips in there. Mm-hmm. Well, for a full plate, we go through 7.5 boxes. So that means that I have to swap out the empty boxes for full boxes several times throughout the run.
Oh, so every 10, 15 minutes you're swapping things in and out.
Yeah, you can so there's some incubation steps where it sits for 10 minutes. Actually, that's the first thing that it's going to do after it mixes with the beads. It's actually just going to sit for 10 minutes. And that's a good time to get up and take a break, go get a drink of water, go
walk around, get some activity.
but yeah, but there are other things that need to be done around the lab. So there's we have shelves full of supplies in the lab, but we're a little limited on our space in the lab. So we have a whole area of supplies up in the office space, one floor up from here on the fourth floor.
Okay.
And so sometimes I'll bring down more supplies. Sometimes I get a notification that there's a delivery downstairs of
Oh,
more
sure.
supplies, and
That's right. Those are getting more towards like
yep,
just the general lab manager.
yep, I'll bring those up. Or we also have all of our waste is all considered biohazard waste.
Oh,
And
right.
at
Yeah.
the University of Florida, all of the biohazard waste gets autoclaved, so
Okay.
sterilized before it actually gets hauled away
Mm-hmm.
by the company that we use to do that.
Right.
And so I'll take some time to pull biohazard waste out of trash cans and stick it in the autoclave
Mm-hmm.
and do that as well.
Yeah. So being a lab manager, is this your main focus because it is so time intensive with the amount of, like you said, you have to do more. You have to just
Yeah,
manage more supplies, worry
right.
about biohazards. Is this kind of your main focus
Yeah, I
now
mean,
for?
right now I spend a good bit of my time here in this lab,
Mm-hmm.
whether I'm running the robot or helping out around um, with other things. We have samples in the freezer that have to be organized and other things like
Sure.
that. So there's always something around that needs to
Right.
be done. And but I do still spend a little bit of time working at home on
Right.
various things um,
So when,
for the lab, for the
yeah.
other lab, for Tara's lab.
So when it comes to looking at, kind of the impact of COVID-19 on maybe like any like workplace wellness activities, like how you feel about your day-to-day job, like how is it, is this kind of just business as usual, or you're just kind of just running a new robot this time, or how has, the current pandemic kind of affected your overall?
Um, I mean, it's a little different. You can stay pretty busy at times, just running around. Things don't go right, and you
Mm-hmm.
got to figure out how to make them go right. But but I mean, I do a mix of working in the lab in my normal daily job in Tara's lab. Do a mix of working in the lab and
Mm-hmm.
working at my desk. Um, I guess a little different. I'm not the one doing any of the ordering of supplies here like I normally do.
Oh, sure,
um,
sure.
but someone else is taking care of all of that. But yeah, it's I will say working at EPI, I actually have stairs here, so I actually spend time
Oh,
going
yeah,
up and down the
that's great.
stairs.
Yeah, that
And
is
I
great.
don't do that when I'm over in my other building because it's flat.
Ah, yeah, that's an interesting kind of angle of getting some good more physical activity during the workday because of
Right.
now your work is taking you to the sixth
Yep.
floor. Or which floor are you on?
Right. I'm on the third floor,
Third
and then
floor.
we have stuff on the fourth floor as well. So I spend a
Okay.
lot of time going between the third and fourth floor, and that's just
Yeah.
not worth taking the elevator for.
Yeah, that's nice. So, okay, so you've been in, and kind of wrap up this this webinar here with just two questions. So you've been testing now for a good 3-4 weeks now. So what do you know now that was kind of like the biggest surprise that you didn't when you first started? What's kind of been like the most interesting thing you've learned through this process?
um, I don't know. I mean, I guess just what all goes into running more of a clinical lab. All the paperwork that we do. We do a lot of, every day we get a worksheet, and every
Mm-hmm.
time I do something, I gotta check it off or write down the time that I did it, which is not quite how it is when you're doing research.
Right.
Um, so I mean, I guess just a lot of what it takes to run a clinical lab, and it takes a lot when you're running this many samples in a day. That's
Mm-hmm.
not something we normally do in a research type setting. You run, maybe 20 samples, and
Oh,
that's
right.
it. Not running 88 times 4 samples
Mm-hmm.
a day.
Yeah. So then, just if I try to paraphrase everything, then I was more of just looking at just the amount of work and consistency around the clock when it
Right.
comes to running samples is a lot
Mm-hmm.
more intense. All right, so these samples are coming from every type of person, right? Symptomatic and asymptomatic, right?
So mostly what we get is symptomatic, but other labs in the area are asymptomatic is what we're getting, but other labs in the area
Mm-hmm.
are getting symptomatic. So like the samples that we received from the villages, I think the symptomatic samples went to a certified lab, and the non-symptomatic
Mm-hmm.
came to us as a research lab.
I see. So there, how many like testing labs are there in Gainesville or North Florida that's like doing a similar type test like you're doing?
Right. I actually don't know. I know there's at least one more at UF.
Okay.
Um,
Is
and
that
I'm not
within
sure
is that
exactly
within public
where
health?
that is. Sorry.
Is that within public health?
No, it's not. I think
Okay.
it might be pathology or something like that.
Yeah.
I'm not sure. Um, and then um, and then I'm sure there's other labs outside of the university in the area that
Mm-hmm.
are, certified labs that are running a lot of the samples from the hospital.
Yeah. Well, I think what you do is it's really interesting, important work.